I'm Dr. Kimberly Stroka, and my lab is the Cell and Microenvironment Engineering Lab here in the Fischell Department of Bioengineering at the University of Maryland, College Park. So I have been a faculty member here since January of 2015. So let me just give you the brief 30-second intro of myself. I grew up in Northeastern Pennsylvania. I received my bachelor's in physics from Denison University, which is a small, primarily undergraduate institution in Ohio. And then I went on to complete my PhD in this department, the Fischell Department of Bioengineering. And I joined the PhD program in the first year the department became a department back in 2006. I then graduated in 2011 and went on to do a postdoc at Johns Hopkins in their Institute for Nanobiotechnology for 3 years before coming back for a faculty position here in 2015. So I know about this department from both the graduate student perspective and the faculty perspective. And there's two things I wanted to say. First is that it has been amazing to witness the growth and momentum of this department over the past 15 years. And second, I will say that the reason I first joined this department as a graduate student is the same reason I wanted to come back as a faculty member, and that's the people and the culture. It was clear and continues to be clear that faculty and students alike are excited about their research and eager to collaborate with each other. And I found it to be really a uniquely positive and supportive environment. So besides that, UMD has many other attractive features, including fantastic resources. And a few years ago, we moved into a brand new building dedicated to the bioengineering department, which has been really fun. The Maryland, DC area is a hot spot for biotech startups, collaborations with the NIH. And it's the type of place where, regardless of whether you like to live in the city, the suburbs, or the country, you can basically find it all. And both the mountains and the beach are just a day trip away. So my lab explores the role of mechanical forces in physiological and pathological phenomena. And to accomplish this goal, we engineer and then probe in vitro models of multi-scale biological systems. And we do this using bioengineering tools such as microfabricated devices. We use novel combinations of cells, molecular biology techniques, and cell mechanics tools. And we also create custom quantitative image processing software. So all of our research falls really into three main focus areas, including exploring the mechanobiology of the vascular endothelium, and especially the blood-brain barrier in the context of health, disease, and drug delivery. Second, we probe cell-to-cell interactions at the blood-brain barrier during tumor metastasis and kind of understand how tumor cells interact with those cells of the blood-brain barrier. And then thirdly, we use engineered cellular microenvironments to understand how mechanical forces alter basic mechanisms of cell adhesion, migration, and division. So currently my lab consists of one research assistant professor, 3 bioengineering PhD students, and 3 bioengineering undergraduate students. And we are also looking forward to having a new MD PhD student join us this summer. So in our recent lab Zoom meeting, I asked each member of my lab to introduce themselves. So.
Hi guys, my name is Becca. I'm a 5th year PhD student. My research project is looking at how physical confinement alters cancer cell division and also cancer cell migration. Why I joined the lab is because Kim is a fantastic mentor and she's really helped me grow as a researcher.
Hi everyone, my name is Ariana. I am a 3rd year grad student here. And my research project is focused on breast cancer metastasis and trying to understand why breast cancer prefers specific microenvironments. I joined the department because I really liked the kind of energy and vibe that I got from a lot of the people that I met here. And I knew that I would have a good chance of finding a great advisor as well.
Hi, my name is Caitlin Kroonert. I'm a junior undergraduate researcher in the Stroka lab. My project focuses on cancer cell stages and divisions, and I joined the Stroka lab because it seemed really collaborative and it seemed like a great learning environment.
Hi, I'm Lee. I'm the research assistant professor in Dr. Stroka lab, and my research focuses on the brain macrovascular endothelial cells. And my study is to investigate how the mechanicals like the stiffness and the matrix shares trace affect the function of the brain endothelial cells. And I joined Dr. Stroka lab because I love the environment here, and I also enjoy to work with her and all the lab members.
Hi, everyone. My name is Adam. I'm a senior undergraduate student here at the University of Maryland, and my project is currently focused on adding some new features to the lab's GenApp software, including cell alignment and mean fluorescence intensity of different proteins, and then eventually running some correlation studies of different parameters under shear stress conditions. And I decided to major in bioengineering because I was really fascinated and excited about the field and its implications and impact on human health and quality of life.
How's it going? My name is Ian. I am a first-year PhD student. I'm just starting out, but I'm doing work in looking into how a protein that regulates the osmolarity of a cell affects cancer metastasis. And I guess the reason I joined the lab, I thought the research was cool, but really it was because of the amazing people.
Hi, everybody. My name is Cole. I'm a junior undergrad, just joined the Stroka lab. Is working to develop a more robust way to analyze cell-to-cell junctions and how that impacts membrane permeability and cell monolayers.
All right, thank you to all my lab members for introducing yourselves.
All right, well, thanks for watching my lab's video, and I hope you all have a great virtual visit to our Fischell Department of Bioengineering.